Welcome to the Cancer Neuroscience Symposium Conversation Series. I'm Monty Montano, Executive Editor of Wiley's Advanced Portfolio. We're excited to partner with MD Anderson and host this series to hear from key speakers featured in the February 2026 Cancer Neuroscience Symposium. Thank you for tuning in as we explore ideas to advance cancer neuroscience together. So I guess we like to start with just learning a little bit about you and sort of how you arrived from Melbourne, I think, to NYU and how you sort of became fascinated with astrocyte biology. So,
Yeah, well, thank you for also being here with me, Monty and Kenzie. It's great to have you here as well. And it's always fun to talk about a topic that you're really passionate about. Um, I guess, yeah, you picked up that I'm not a Native American. My accent is not from Brooklyn. I am Australian. I actually grew up on the west coast of Australia, so almost the direct antipode of New York City. But my academic journey is a little winding path. I desperately wanted to be a medic when I was in high school. At some stage, I realized I'd probably have a horrible bedside manner. So I actually went to Melbourne, as you correctly mentioned, and studied physics and chemistry. And then I actually had brain surgery myself and got really fascinated by the brain. And so switched degrees and actually started studying anatomy, cell biology, bioinformatics, and then did my PhD in pharmacology. Was very fascinated in the blood-brain barrier and how do we get drugs delivered to the central nervous system in a way that could treat patients that have neurological conditions, but also from a perspective of a pregnant woman, how can she take drugs that she may need that could be toxic to her child, unborn child, and vice versa. If the child needs drugs, but we don't want to give to the mother's brain, this was something that I was really, really interested in development. And then I moved to America. I moved to Stanford about a decade and a half ago to do my postdoc in the lab of the late, great Ben Barres at Stanford. And from there, things completely changed for me. I was still very much interested in the brain and in pharmacology, but rather than focusing on drug penetration, I was really focused on glial cells, particularly astrocytes and neuroinflammation in the context of a whole host of different neurological conditions, including, of course, how these glial cells respond to and interact with cancers of the brain.
That's just fascinating. How like something like brain surgery could just sort of pivot your whole approach, uh, and it makes me think about your work on sort of how astrocytes themselves become reactive to their life experiences as cells, and perhaps you could speak a little bit to the sort of I think the literature that you're one of the founders of this sort of astrocyte reactivity and what that involves.
Yeah, and maybe I should take a step back as well. So for those of you who are not spending your entire life like me focused on astrocytes, what is an astrocyte? So like most things, named by some ancient Greeks. So astro means star, sorry, and cyte means cell. So it's a star-shaped cell. Although now we think of more of them like a bushy sponge or something like this in the brain, and they are a non-neuronal cell, a glial cell. There are many glial cells in the brain. There are glial cells that wrap around neuron projections or axons to make sure that your electrical conduction velocity is very fast. There are glial cells that are involved with the blood vasculature, like pericytes. There are microglia, which are resident immune cells of the brain. And then a large component of the glial cells are actually astrocytes. And these are really important supporting cells of the brain. So I like to think of them as the mothers of the central nervous system. They provide all of the nutrients to the teenagers of the central nervous system, which is the neurons, loud, rambunctious, making lots of noise, but generally poor at looking after themselves. So astrocytes provide all of the nutrients to these neurons, and they take away all the metabolic waste products so that neurons can function really efficiently. And so astrocytic functions are integral for every species in which we found glial cells like astrocytes in for the brain to function normally, and the spinal cord, but the central nervous system. And unfortunately, in the context of acute infections or traumas or chronic disorders like neurodegenerative diseases or cancer, these astrocytes take on a response that we call reactivity. They're reacting to something external, and they change their functions. And in some instances, they become extremely extra supportive of these central nervous system cells like neurons to make sure that they can keep functioning in these adverse conditions. But in sometimes they take on a detrimental function that can actually lead to an exacerbation or a fastening or a quickening of the degeneration that's occurring. And these two polar states, they're not only two states, there are of course many, many different heterogeneous states that many labs all around the world are discovering, which is really exciting. But they interact in these sort of two basic good versus bad, or there'll be maybe multiple good and multiple bad. And so what my lab is really interested in is how do those glial cells interact with other cells, be those cancer cells or neurons or the blood vasculature or immune cells. So that's sort of like our immunology hat. We're also very interested in what is particular molecules that either drive their induction of different reactive states or drive positive or adverse reactions in those cells. This is our sort of biochemistry pharmacology hat from my PhD. And then we learn a lot across diseases. We may study things in the context of, say, a viral infection or a trauma or glaucoma or Alzheimer's disease, and then we use that to understand what might be happening in these seemingly unrelated diseases to sort of say, is that same mechanism engaged in, say, brain cancers? And that's sort of our putting off our oncology hat here. And then ultimately we take all of that, we put it together and say, how do those cellular and molecular interactions across diseases sort of change the cognition and behavior of either a person or a patient? And that's sort of our psychology, sort of behavioral neuroscience hat that we put on, always under the guise of what is an astrocyte doing as a nexus of this interaction.
I mean, I think it's really, really fascinating. And Kenzie, chime in when you've got that burning question. But this discussion of sort of the toggling of a good, the good guy, bad guy.
Mm-hmm.
It makes me think that there's a certain amount of plasticity in astrocyte function. And I just wonder, are these different subsets of astrocytes, which I'm sure there are lots of subtypes and subsets, or is there an actual plasticity of astrocytes so that they change their function depending on the microenvironment or perhaps even the macroenvironment?
This is my whole reason for being. This is such a great question, and I think I could rattle off dozens of names of amazing scientists that have investigated this over the last few decades. And there's this. You said both subtypes and substates. I think this is a really important way of thinking about this. So we think about heterogeneity, the sort of diverse population of, in this context, astrocytes, but it could be neurons, it could be cancer cells, it could be anything. But we'll contextualize this in astrocytes. We think about this as a heterogeneous population of cells under normal physiological conditions. So we know, for instance, from work from people like Baljit Kark at UCLA, that striatal astrocytes are very different to cortical astrocytes, and hippocampal astrocytes are very different to thalamic astrocytes and things like this. We also know from work from Omar Bayraktar, who's in the UK, that there is even diversity of astrocytes in different cortical layers of the brain, much like we see with neurons. So there is this sort of regional heterogeneity that exists. But there's also developmental heterogeneity too. So people like Stephen Sloan, who's at Emory, and Nicola Allen at the Salk Institute have really looked at how is the gene expression profile or the proteomic profile of these astrocytes changing from an intrauterine environment through childhood, through adolescence to adulthood and aging, and how would that change the way that these astrocytes in those different brain regions at different stages of life have different functions that could be really important for the microenvironment that they find themselves in at that particular time in that particular region. So that's all itself already very complex. And then we add on top of that the fact that this microenvironment changes in the context of all these different pathologies like cancer. So then we're starting to think about, well, how does the heterogeneity of an astrocyte's function change in that particular brain region at that particular time or age in that particular region of the pathology? So if we're talking about a tumor, in the center of the tumor versus the outside of the tumor versus further away from the tumor versus a region that may be connected by a neuron to that tumor, et cetera, et cetera. So the heterogeneity problem, just the sheer complexity of this heterogeneity problem is, I think, really exciting and also terrifying to some degree, because we have to really take down this idea of one type of astrocyte or a good versus bad astrocyte to multiple states that are interacting with one another, and they're changing in the context, both spatiotemporal context and disease context as well. So it's wonderfully heterogeneous. And you sort of touched on there, is it plastic? I would say yes. We have beautiful evidence that reactivity mostly resolves with time. But if the drivers of that reactivity persist, as happens in, say, a tumor or happens in, say, around some amyloid deposit in Alzheimer's disease, then that reactivity can persist. And so the question is, can we therapeutically target that reactivity, or do we need to target the underlying pathology? And what is this, the chicken or egg argument? Which is the best way to approach this therapeutically? My guess is probably co-therapeutically targeting both. But this is a really good question.
Very, very, very cool, and obviously very, very complicated. I'm just thinking about like how does such a complex system maintain homeostasis? I mean, with all these interacting parts and exposures and the trajectory of exposures, what does the astrocyte say to itself and its neighbors and its partners in the brain? Let's reset. We've gone too far along this pathway. Let's rethink this, so to speak, and find our homeostatic balance again. I mean, how is that achieved, or is it something that is just sort of lost in the history of exposures and responses?
Oh, this is also great. Actually, this is a project that an MD PhD student in my lab, Amy Guo, was actually investigating now. And so I think this I think is almost the holy grail of reactivity. Of we sort of know a lot about what makes many different substates of astrocytes become reactive, right? We know there's an interaction with immune cells, we know there's an interaction with vascular cells, an interaction with pathology and cancer, things like this. We know what induces many substates of reactive astrocytes. We have less knowledge about what tells them to turn off again. And so there's two possibilities here. One is this is completely cell autonomous, meaning this is completely a decision. Like you say, the astrocyte says, okay, that's enough of me being reactive. Let's go back to a normal physiological state because that was really good. That's probably slightly personifying an astrocyte more than it deserves. I like to think of astrocytes as being really great responders to what's happening around them. I'm not sure that they're really good initiators of things, but they're really good exacerbators of things. They're like a megaphone. A tiny voice from some pathology or an immune cell gets exacerbated by astrocytes and gets amplified and spread further. And the reason that we think about this is astrocytes are everywhere. They completely tile the central nervous system, the brain and the spinal cord. They are connected by gap junctions that sort of pass signals between astrocytes across long states, a long range of the central nervous system. And this is some work from Melissa Cooper, a postdoc in my lab. And so we know that they have this ability to communicate with one another by responding to these external stimuli. And so what makes them stop being reactive? My guess is maybe not themselves, although there is an argument that it could just be a clock. The reactive clock gets turned on and it counts down and then you're no longer reactive. And if the pathology is still there, you start the clock again. But there's some really good evidence that is an additional external cue. So one external cue turns them on, makes them reactive, and another external cue turns them off, or is the absence of that cue that was there to turn them on, the absence of the cue makes them turn off over a certain amount of time. And so Amy, like I said, Amy Guo in the lab is really digging deep onto this for one particular state of reactive astrocytes that we find that's associated with pathologies and cancers and infections and things like this. But is it the same for every substate? Great question. Kenzie, Monty, do you have answers? I'd love to know the answers. That would be really great. That would save us some time.
No answers.
Well, we're looking forward
questions.
to Amy, presenting me perhaps some of this data at the conference for sure. Kenzie, you were going to say something?
Oh yeah, I wanted to talk more about astrocytes not being good initiators. So thinking about the role of microglia, I think there's a lot of crosstalk there. And normally the way we think about it is astrocytes being what's secreting complement, for example, to modulate microglial function. So what do you think the role of astrocytes in not just direct astrocyte to tumor communication, but as a mediator between neurons or microglia, other cell populations that also talk to the tumor?
Yeah, I think this is a really great question, Kenzie, and I think I'm always I always like to tell people I'm a scientist, not working in the space of religion. So if someone comes with some new data, I'm happy to spin on a dime and change my opinion completely, which is really great. And my trainees often do this for me, which is great. But I should say, oftentimes when I say astrocytes are not a very good initiator of responses, what I tend to say is there's not a lot of conditions in which it is astrocytes that, because of some internal mechanism, become reactive and then spread this reactivity. Of course, there are diseases that have mutations in astrocyte-specific genes. Alexander's disease is a really good one. It's a mutation in this cytoskeletal protein GFAP. We know there are a lot of disease-associated single nucleotide polymorphism SNPs, or mutations that are associated with increased risk of disease. So things like LRRK2 and Parkinson's disease, or ApoE isotype in Alzheimer's disease. So definitely there is internal machinery and changes that can change the way that astrocytes respond to these sort of external stimuli. And so we in the lab start to think about these as being disease astrocytes, ones that cell autonomously internally changed, and reactive astrocytes that are reacting to something externally. You are, however, 100% correct that astrocytes initiate a lot of interactions with cells. So we talked a little bit at the top of the call about interactions with neurons. So we know that astrocytes provide nutrients to neurons, they take metabolic waste products away, they secrete synaptogenic molecules. There's a lot of work of Charlotte Eriglu at Duke that can make synapses, work from Mark Freeman's lab at the Volum, or at Wonsof Chung's lab at the KAIST to show that astrocytes can phagocytose debris in synapses. They do a lot of stuff that they initiate themselves. And of course, they release molecules that can modulate immune function. And like you correctly said, with tumor cells, and they can modulate the way that a tumor interacts. Are astrocytes required for the tumor neuron synapses that form, right? I think this is a really interesting question. We know astrocytes are really important for synaptogenesis. Is that only neuron synapses? I would love to know this. If someone does know this and I haven't read it, you should correct me. And I think this is really cool, because this would give us the ability to maybe have a new therapeutic to stop that from happening, because we know that innovation is important for tumor growth and expansion, things like this. So there's lots and lots and lots of functions of astrocytes that we know are really important. One of the difficulties, to sort of bring it back to the discussion we're having with Monty before, is when we think about this heterogeneity of reactive astrocytes, we don't have a lot of tools at our disposal as a field to understand or to go in and probe exactly what the functional changes are in these substates of cells. Sequencing is kind of easy. It is lowering in cost, and it's simple in inverted commas for us to say, here is 6 or half a dozen or a dozen different transcriptomically defined reactive astrocyte substates, or we can do this proteomically or lipidomically. But what has been a little more complicated is, because of the fact that they are very heterogeneous and integrated with one another in vivo, it has been difficult for us to say that function that's changed is that subtype of astrocyte or that subtype of reactive astrocyte. And so we, at least in my lab, have sort of had a lot of success in taking those transcriptomic profiles or barcodes of substates of reactive astrocytes that we find in vivo, either in patients or in rodent models, and then recapitulating that in a cell culture dish so that we have now a homogeneous population of astrocytes that we can say, now, normally you do this, do you still do that? And then they're just binary questions. Do you still provide trophic support? Yes, no. Do you still make synapses? Yes, no. Can you still phagocytose? Yes, no. And these then become very simple questions that let us build up this understanding of the functional diversity of those astrocytes so that we can actually address, this is a very long-winded answer to your question, Mackenzie, we can actually address your question, which is, do they initiate stuff that is really important for interactions with tumor, for tumor expansion, for tumor inhibition, for all these sorts of things. For metastatic cancers, does it sort of mediate changes in their ability to stick and grow and things like this? And we tend to find, again, all of these same sort of mechanisms that are being engaged in neurodegenerative disease and in acute neurotrauma and acute infection are being engaged to some degree in these cancers as well. And I think that means that we have this ability to sort of take these sort of disparate disease states to sort of better understand what might be happening in these cancers.
You mentioned tumor inhibition and right after commenting on heterogeneity, and I'm just thinking about, obviously immunotherapy is a big question, and how immunotherapy is effective and off-target effects of immunotherapy. And so maybe speak a little bit to the role of astrocytes in immunotherapy and whether this heterogeneity is both a problem and an opportunity, and whether that can be dialed down to specific subsets or context, especially like CAR T immunotherapy.
Yeah, no, I think this is the holy grail. The holy grail would be you have a drug that inhibits or promotes the formation or reversal of every substate of reactive astrocytes that anyone around the world has been able to define. And then you sort of say, in this particular disease context, with this particular patient genetic predisposition at this particular time, this is the treatment that we give. Because there could be an instance, and a perfect example I use is some work we did with Aaron Gibson, who at the time was a postdoc in Michel Mongi's lab when we were all contemporaries at Stanford, is we discovered this subtype of neurotoxic reactive astrocytes. We've now since shown that they're present in lots of different neurodegenerative diseases, and they secrete a toxic lipid. This is some work that Kevin Gutenplan did. And we were like, oh my God, this is really bad. We want to turn this off. We want to make a therapy that turns this substate off because it kills neurons. And killing neurons is inherently bad because that is neurodegeneration. And if we can stop that from happening, that'd be really great. But what Aaron had found, we were in buildings adjacent, is that actually treatment of tumor models with methotrexate would actually mount an immune response, which is what we see in lots of different things. That immune response was mounting this neurotoxic response in astrocytes. And then that neurotoxicity was able to kill the tumor cells. And so here we have an instance where we have inherently a quote unquote bad reactive astrocyte that is actually arguably doing something that is really good. And so now you have this sort of additional layer of complexity that what we think about what this cell is doing, which in the context of your brain, my brain, would be really, really bad. In the context of this particular disease, actually this is a reactivity subset that we would want to drive, because we would want to be able to kill the tumor, and that is essentially using this really important sort of internal response that the central nervous system has that is essentially fighting and staving off this tumor, and that would be really, really good. But we have to titrate it to make sure once the tumor is all gone, we're not killing neurons nearby that could be equally susceptible, et cetera, et cetera. So I find this like the most exciting and compelling question to keep doing what we're doing, and the entire neuro-oncology field to keep doing what they're doing and talking really closely with neuroimmunologists, because this is not an insurmountable problem. All we just need to do is understand the heterogeneity, which we've talked a lot about, but also understand the timing of when we may want to turn on or turn off a response. And it may be that there is a therapeutic that early in disease or early in cancer diagnosis, you want to exacerbate this response, and then later on you want to reverse or turn that response off. And this is the sort of fidelity that we're now turning towards in the lab, because this may be the best chance that we have to sort of modulate reactivity on the astrocyte side, which of course, as Kenzie pointed out, would also modulate the immune response as well, and they go hand in hand. It's no longer a chicken or egg argument. It's sort of like, it's never the chicken or the egg, it's always the lizard, right, that comes first.
I think it's absolutely fascinating. I just love this thing. It reminds me of tissue remodeling in response to injury, how you have different subsets of macrophages that do ostensibly different, they perform different functions, and perhaps some of the problems are associated with persistence of one particular function versus another, like pro-inflammatory versus anti-inflammatory. And so it's not just turning off the anti-inflammatory. They're important for recognizing and sort of clearing out debris. The other scenario that this reminds me of is in like senescence. There's a lot of effort in the aging field to turn off senescence and senescence biology, but senescence is pretty important for, many sort of functions, from wound healing to regeneration and axolotl. And so it's this idea of fidelity and being able to not label something as the bad guy, but recognizing that sometimes that function is essential, and it's really more sort of tuning in and tuning off is, I think, a sort of a very sophisticated way of thinking about this. So I'm very excited to hear more about your progress in this line of thinking at the conference. So this is super cool.
Yeah, we picked a really interesting problem. We made it more and more complex with everything that we discovered. And there's so many amazing people, not only in the neuro-oncology field, but in the astrocyte field, the neuroimmunology field. And together, we're all sort of adding in these really important pieces of this pretty complex biology and sort of building out this puzzle. And this has really only been possible, there's been a lot of people working in astrocyte biology for decades and decades and decades, but we've always sort of lagged behind a little bit in the tools that are at our disposal. And it's really been in the last, decade and a half, 2 decades, that people have been developing all these amazing tools, the ability to culture astrocytes so that they retain their in vivo gene expression profiles and functions so that we can use them for high throughput screening for drugs that are FDA approved, so we can use them off label or to screen for new drugs and compounds, which is really, really powerful. People like Amy Gleichman, who was at UCLA, and Tom Carmichael's lab is now based at The Ohio State. She's made a bunch of viral tools to actually target specific populations of astrocytes, so we can turn on and off genes and look at what's happening in specific regions. These tools are just coming online now. So it's really, really exciting to be in this space, because all of the questions that everyone has always asked, people way smarter and way more understanding of this field than I've been asking for decades and decades, we can now sort of start to really address them, which I think is really exciting and compelling. So definitely watch this space. I see lots of things coming really quickly on the horizon, which is, I think this is awesome.
Excellent. Well, I can't imagine a better place to pause, and unless there are further questions by Kenzie. I mean, this is just a very uplifting idea of thinking about cognitive resilience and plasticity and the opportunity for therapies that take advantage, not in a bludgeon kind of a fashion, but more fine-tuned and sophisticated, to recognize the important and timely functions of astrocyte subsets and subtypes. Can you speak maybe a little bit in closing to cognitive resilience and sort of how your work fits into this sort of larger goal of building a sort of a resilient brain, I guess?
Yeah, I think this is a good point. This is something that we don't investigate specifically in the lab, but it's something that I think that we think a lot about, just general cognitive function. I mentioned a little bit, we have the behavioral output and the psychological output of everything we do has to leave the brain better than we found it. It's sort of like hike in, hike out, take your trash, right? So this is what we're always thinking about in the context of therapeutic intervention for astrocytes. We don't want to revert the astrocytes to a state that is different for that context and time. And you brought up just earlier, the idea of senescence. We know that astrocytes change with aging, and we don't know that that is inherently bad. Nicola Allen had done this a number of years ago. Laura Clark, who was a contemporary of mine in Ben Barris's lab as well, had investigated this. There's lots of other people investigating this more recently. We had at the time sort of as a field interpreted this that, there's an upregulation of a lot of transcripts here that we associate with reactivity, and so therefore this must be bad. But we don't actually know that that is bad. It's just our interpretation given our understanding of what happens in the young adult healthy brain. And it may in fact be that the way that those astrocytes are in those senescent brains is really important for that microenvironment, bringing it way back to what we started about, was the microenvironment interactions with astrocytes. And so I think that moving forward, we're going to have to think more more carefully about this and say, we don't want to revert an astrocyte to, what it looked like when I was 20. Maybe that astrocyte is not great for my middle-aged brain, and what I need is a middle-aged astrocyte. I could be completely wrong, and maybe we do want to all have 20-year-old astrocytes and we'd be superstars and we'd be able to take on the world. We just don't know that right now. But people are starting to do these transplant experiments, and there is some evidence that this can lead to some basic cognitive benefits in rodent models. So, putting human astrocytes into mice can lead to some cognitive benefits and things. This was done by Steve Goldman a number of years ago now. And so there is this idea that putting stuff that wouldn't normally be there can improve neuronal function. So the idea is putting stuff back to a different state. Could that be beneficial in the recovery phase following these sorts of cancers and diseases? And would that enable us to have a better cognitive function in those stages post disease? And I think that's something that there's not a lot of people investigating, at least on the astrocyte side, and I think it's really compelling. And I would love to sort of dig deeper and scratch this a bit, because we have some tidbits of information that suggest that we might want to play around with it, but we also have tidbits that says maybe we don't want to play around with it. So again, I keep saying watch this space, but watch this space.
Right, absolutely. And you mentioned the 20-year-old astrocyte versus the 40-year-old astrocyte, and it reminds me of this idea of accumulation of experiences and whether they're good and bad and useful or not useful, and the counter efforts these days, at least in the aging space, to partially reprogram and to remove some of that memory, like imprinted memory in cells. And I just wonder, what your thoughts are in terms of like imprinting in astrocyte function over the life course, and the idea of reversing, reprogramming to bring astrocytes back, is that a bad idea? Or or not? This,
Yeah, I don't know that. I definitely don't have enough information to answer that question with a coherent answer. I think important. I definitely know my limits here. I think that there is, there's a lot of work that has been done to show this idea of these senescence differences,
yeah.
right? Rusty Gage has also shown that you can make these immediately differentiated astrocytes from fibroblasts, which are very different from astrocytes. These are human astrocytes that are produced from pluripotent cells as well, right? They have different gene expression profiles. They arguably have some different functions. And so the idea now of, if you were to replace the same region with the same region, but just turn the clock back a little bit, would that be really beneficial or detrimental? Or if you replace, say, an astrocyte from a cortical space with an astrocyte from a hippocampal space, would that be detrimental as well, right? And we don't know enough about whether or not astrocytes would just, is it nature or nurture for an astrocyte phenotype? Are they responding to what's locally there, just the microenvironment? They kind of don't care. If you put a hippocampal astrocyte into a cortical astrocyte, it becomes a cortical astrocyte, not a hippocampal astrocyte. Or if you put an old astrocyte into a young brain, or vice versa, does it just become what it is in around? That's possible, and there's definitely some work that has shown that happens to some degree. But there's also some work that has shown that they have this sort of retention of that gene expression barcode that made them what they were, where they developed in the first place, that they retain as well. And so I think that we can tweak with this a little bit as we move forward. And I think that's going to be a really compelling space that people, once again, now that we have the tools, that people are going to start playing around with this and say, is there something that changes in the context of being close to a tumor that means they're always going to be a close to a tumor astrocyte, and would that be detrimental? Or can we change them to be an astrocyte that thinks it's never interacted with a tumor before?
That's fascinating. Even after the tumor may be gone, it still thinks and behaves as though the tumor were adjacent. That's fascinating. Well, I mean, congratulations on being in such an extraordinarily interesting space at this time in sort of the development of cancer, understanding cancer in microenvironments and therapeutic interventions. Kenzie, any last thoughts before we say we're looking forward to seeing you, Shane, in February with everybody else in Texas?
I have plenty of questions, but I'll ask them in February.
That sounds fantastic, and it's always fun to be in Houston, so I'm not going to argue that it'd be a bad thing to sit down for a few hours and talk about neuro-oncology and astrocytes.
Yeah. Well, Shane, thank you so much for taking some time to chat with us. We very much look forward to reading about your work, more of your work, and to seeing you in Texas in a few short months. And otherwise, just thank you again for all the time and your great work.
Thank you so much. This has been great, and you've planted some seeds for me to think about more. So hopefully I have answers for both you and Kenzie early next year.
Excellent. Okay. Thank you. Thanks for tuning in today. We hope you'll join us as we continue the conversation in Houston and virtually at the 2026 Cancer Neuroscience Symposium hosted by MD Anderson and Wiley's Advanced Portfolio.